Hey guys, today I'm going to talk to you about protein synthesis. It is a process in which cells make proteins. There's two stages. The first one is transcription, transfer of genetic instructions in DNA to mRNA. The second stage is translation, which is when the mRNA along with the tRNA and the ribosome work together to make a protein. We see this in the process of decoding. Decoding is a process from DNA to mRNA to tRNA and then to finding the amino acids in the strand. DNA is made up of adenine, cytosine, guanine, and thymine. In this case, adenine will bind with thymine and cytosine will bind with guanine. mRNA, on the other hand, is made up of adenine, cytosine, guanine, and uracil. In mRNA, adenine will bind with uracil and cytosine will bind with guanine. The only difference between mRNA and DNA is that the mRNA has uracil and DNA has thymine. Okay, in this example, our DNA strand will be TAC, GCA, TGG, and AAT. The complementary strand will be ATG because thymine binds with adenine and cytosine binds with guanine. So then we have CGT, ACC, TTA. The mRNA strand, remember, has uracil instead of thymine. So adenine will bind with uracil, and then we have T to A, G to C, and so forth, GCA, UGG, AAU. In this example, the mRNA strand will take the strand out of the nucleus and into the ribosome, and this is where translation occurs. Then, tRNA strand to the mRNA strand will be AUG, CGU, ACC, UUA. It will bind next to the mRNA, and this is where transcription occurs. You will then use the tRNA strand to find the amino acids. Every 3 letters make up what we call a codon, which carry a particular code for an amino acid. 1 codon, 2 codons, 3 codons, and the 4th codon. You will then use the amino acid chart to find the amino acid to every codon in the strand. So, the chart will look something like this. There's one that will have a circle. In this case, we'll use the square. Okay, so we will start with the first codon, AUG, which we find here is methionine. Then we have CGU, which is going to be CGU, arginine. Then we have ACC, which is threonine, and our last one is UUA, which is leucine. Now, there's things called mutations, which I will talk about briefly. In this case, if a letter changes here and it changes the amino acid to serine, it would be called a missense mutation. If this letter changes but there's no change to the amino acid at the end, it would be called a silent mutation. I hope this helped with the understanding of protein synthesis as well as decoding. Thank you guys.